This is the cell cycle of animal cells. The cell cycle begins with interphase, where it spends 90% of the time before mitosis can occur. Interphase has 3 stages within it. The first stage is the G1 phase. During the G1 phase, the synthesis of RNAs and proteins occur. The cell also grows while carrying out specific functions that the cell type requires. It also makes copies of organelles. The second stage is the S phase. In the S phase, DNA replication occurs and the cell continues to grow. The third stage is the G2 phase. During the G2 phase, the cell finishes making copies of the organelles and prepares to divide. Now cell division can occur, allowing mitosis to begin. The first phase of mitosis is prophase. In prophase, the sister chromatids are condensed into chromosomes. The nuclear membrane begins to break down and the nucleus disappears. Spindle fibers, which are microtubules made up of proteins, grow from the centrosomes and attach to the kinetochores on the centromeres of the condensed sister chromatids. Asters, which are the microtubules from the centrosomes, push out the cell sides, creating a larger cell. The second phase is metaphase. In metaphase, the spindle fibers from the centrosomes move the chromosomes to the metaphase plate at the center of the cell. The centrosomes have migrated to opposite sides of the cell. The third phase is anaphase. In anaphase, the spindle fibers retract, separating the sister chromatids, leaving complete and equal sets of chromosomes on opposite ends of the elongated cell. The fourth phase is telophase. In telophase, the nuclear envelope begins to reform, the chromosomes begin to relax, the nucleolus reappears, the spindle fibers break down, and the cell begins to pinch inward. The fifth phase is cytokinesis. In cytokinesis, the cell pinches inward until the cell is completely split into 2 identical daughter cells. A cleavage furrow is formed, caused by active filaments in the cell. There are now 2 cells, each genetically